जी मैम।
जेनेटिक कोड डीजेनरेसी क्या होता है?
रिपीट करें मैम, ये आवाज का थोड़ा सा इशू।
जेनेटिक कोड डीजेनरेसी क्या होता है?
मीन से अच्छा जीन कोड, ये अच्छा उसकी डिजेनरेसी। जी जी। अच्छा मैम, उसमें ये होता है आप अमाइनो एसिड्स, देखिए मैं इसको कोड कर।
आवाज नहीं आ रही आपकी, समझ नहीं आई क्या कहा है आपने।
अच्छा मैम, मैं ये कह रहा था कोड जो डिजेनरेसी होती है उसमें बेसिकली एक जो आपका वो होता है अमीनो एसिड होता है, उसको जो है मीन्स डिफरेंट जो जेनेटिक कोड्स होते हैं, दो या दो से ज्यादा, वो जो है एक ही अमीनो एसिड को कोड कर रहे होते हैं, तो उसी को जेनेटिक डिजेनरेसी।
इसकी रीजन क्या होती है? आसमा जफर, आप बताएंगे कि क्यों क्यों इस तरह होता है, जेनेटिक कोड डीजेनरेट कैसे हो जाता है, क्या रीजन है इसकी?
मैम हम इसमें ये कह सकते हैं कि जो डीजेनरेसी है कोड की इस वजह से होती है कि जो 3 न्यूक्लियोटाइड्स होते हैं कोडॉन के, उसमें जो फर्स्ट 2 न्यूक्लियोटाइड्स हैं वो वाटसन क्रिक को वे करते हैं, लेकिन जो तीसरा कोडॉन है उसमें फ्लेक्सिबिलिटी होती है वूबल बेस पेयरिंग की, तो इस वजह से डिफरेंट कोडॉन्स जो हैं वो सिंगल अमाइनो एसिड को कोड करते हैं।
आपका काफी हद तक आंसर ठीक है, लेकिन इसको और थोड़ा सा रखें ना कि जो एंटी कोडॉन में जो की जो फर्स्ट बेस होती है, उसमें आपके पास काफ़ी रूम है ना, कि अगर उसकी बेस जो है वो आई है या फिर ए है, तो उसके कॉरिस्पॉन्डिंगली आप ये कह सकते हैं कि वो फिर कोडॉन में जो उसकी बनती है थर्ड वाली, वो उसके पास चॉइस ज़्यादा आ जाती है ना फिर।
यस मैम।
ठीक है ना, तो वो जो थर्ड बेस पेयर के अंदर आपके पास एंटी कोडॉन, फर्स्ट बेस पेयर जो एंटी कोडॉन का है उसके अंदर, और थर्ड बेस पेयर जो कोडॉन का है, यहाँ पे आपके पास एक तरह से लूज या वोबल किस्म की बेस पेयरिंग है। ठीक है, तो उसकी वजह से आपके पास फिर वैरायटी आ जाती है। ठीक है, और यही रीज़न है कि जब आपके पास अगर जिनोम लेवल पर, डीएनए लेवल पर अगर कोई म्यूटेशंस आती भी है ना, तो वो ज़रूरी नहीं है कि अमाइनो एसिड को चेंज करें। ठीक है, वो आपको फिर देखना पड़ता है कि किस टाइप की म्यूटेशन है, और अगर वो थर्ड वाले उसमें आ रही है तो फिर चांस है कि वो शायद कोई चेंज नहीं करेगी उसको, अमाइनो एसिड को। ठीक है,
जिन्हें हम नॉनसेंस म्यूटेशन कहते हैं ना।
हाँ जी, वो भी कह सकते हैं नॉनसेंस। ठीक है, सेंस नॉनसेंस म्यूटेशन आ रही है। ओके, सो आप लोग को सबको नज़र आ रहा है ये ट्रांसलेशन इन यूकैरियोट्स? शेयर हो रहा है ना ये आपके पास?
यस मैम।
Okay, so now we will be studying about translation process in eukaryotes. We know that in eukaryotes, 80s eukaryotic ribosomes are found actually, right? And they are bigger than the 70s prokaryotic ribosomes. So in total, the small subunit and large subunit, small subunit is 40s and large subunit is 60s in size. And the 60s subunit contains 28S ribosomal RNA, 5.8S, and 5S ribosomal RNA. And the small subunit 40S, that contains 18S ribosomal RNA. And you can see the structures of E. coli and then in comparison with yeast, large and small ribosomes. So in comparison with prokaryotes and eukaryotes, you can see the differences here in the case of ribosomes. You can see the large subunit has 28S ribosomal RNA in place of 23S ribosomal RNA that was found in prokaryotes. And then the small subunit was having 18S ribosomal RNA in eukaryotes, and in prokaryotes it was having 16S ribosomal RNA. Okay, so the functions of these RNAs would be almost same as we have studied in case of prokaryotes. Okay, And then you can see the differences in the number of proteins in the large and
मैम आवाज नहीं आ रही।
मैम आपकी आवाज़ नहीं आ रही। रिकॉर्डिंग स्टॉप कर दीजिए।
मैम के मैसेज आया है कि लाइट चली गई है।
in eukaryotes. The first step is again the same, that ribosome, they have to recognize and bind the messenger RNA, right? So this binding is basically different in case of eukaryotes. The ribosome binds to the 5' cap and scans down the message until it reaches the first AUG, okay? So you just remember that in case of prokaryotes, the situation was different, right? There we were having Schindler-Garno sequence, that was the ribosome binding site. And here in case of eukaryotes, we are having 5' cap, right? So this 5' cap, again, just recall the previous information, is very important for the translation process as well, okay? So, and all of you know that majority of messenger RNA produced in eukaryotes, they have their 5' modification that is called as cap. And this is used for the recognition of ribosomes to start the translation process, right? So we also know that the messages in case of prokaryotes, they are polycistronic. It means that single messenger RNA was actually encoding more than one proteins, and that these proteins were translated. While in case of eukaryotes, although generally it is believed that the eukaryotic messenger RNA, they are monocistronic, but in last 4-5 years, many scientists actually have explored that eukaryotic messenger RNA, they can encode, the single messenger RNA, it can encode more than one protein. It means that the open reading frame that will be including start codon and stop codon, there could be many open reading frames in the single messenger RNA, okay? So there is increasing evidence is actually for this thought actually, okay? So, although in previous texts you would be finding that messenger RNA are monocistronic, okay, but the latest information in knowledge actually tells us that eukaryotic messenger RNA, they do have additional, they can encode additional proteins, okay? They can have more than one open reading frames. So translation begins with methionine again and involves a special initiator transfer RNA that is called as methionine tRNA I, okay, for initiation, for initiator. But the methionine is not formulated in eukaryotic system, okay? So this is one of the difference. In case of prokaryotes, methionine was formulated, while in case of eukaryote, it is not formulated, but special initiator transfer RNA is designated to bring the initial methionine to the start site. So in eukaryotes, ribosomes bind to 5' cap, and then they move down the messenger RNA until they reach the first AUG, the codon for methionine, and translation actually starts from this point. Eukaryotic messenger RNA, the codon for single gene, but definitely there are exceptions, right? So note the translation does not start at the first base of the messenger RNA. There is untranslated region at the beginning of the messenger RNA, and that we call it 5' UTR. Okay. So here are the steps in the initiation process. The eukaryotic initiation complex, actually you will be seeing that the eukaryotic initiation and elongation, they are quite different from the prokaryotic one. And although we do have similar kind of proteins which perform the similar kind of function, but the number of proteins can be larger or higher in case of eukaryotes. So you can see that here messenger RNA is shown in pink, and then there's yellow circled cap is shown at 5' end. And so the eukaryotic initiation factor 2 along with GTP, that will be actually for start AUG only, that will be actually bringing the first amino acid methionine to the first transfer RNA, having the methionine to the start site. Okay, and definitely this will not be formilated. And then the small subunit of the ribosome will be placed and will be having interaction with the messenger RNA and the first transfer RNA. And then there will be the involvement of large subunit, okay, 60S subunit, and that will be requiring the GTP, okay, and then will be the elongation process. So now let's see the step by step in the initiation process. So first is the capture of messenger RNA. The 5' cap, that is M7G PPX of the messenger RNA, is captured by binding to a complex of eukaryotic initiation factors of the family EIF4. Okay, so this is the whole family, and this family contains many members like A, B, G, and E. And these members actually they bound, they recognize and hold the messenger RNA. And then other members like 4A and 4B, they unravel the kinks in the messenger RNA, and collectively they are called as eukaryotic initiation factor 4F. Okay, so let's see in this figure, you can see that on the top messenger RNA is shown, and in the central region you can see the kink or the secondary structures. You know that messenger RNA in case of eukaryotes, they are quite large, and then they can have secondary structures in their whole structure. So these secondary structures should be removed actually. Okay, so these initiation factors, they not only recognize the messenger RNA, but they also remove the secondary structures within the messenger RNA. Okay, which might exist before the AUG, and then they and with the utilizing the ATP, they are actually doing this job. So eukaryotic messenger RNA, so you can see here the general picture that at 5' end we are having initiation factors, whole family, and at 3' end we are having poly A. Okay, remember the poly A tail at 3' end. So this poly A tail is recognized by poly A tail binding proteins. So here they have mentioned it as PABP, right? So this is the whole family of proteins which actually bind to this poly A tail, and then protein and protein RNA interaction actually circularizes the messenger RNA. So what does it mean that these messenger RNA, they get circularized? Because the proteins which are bound to the 5' cap and the protein which bound to the 3' tail, they interact with each other as well. So hence they are actually circularizing the messenger RNA. So actually the phosphorylation and dephosphorylation of these initiation factors and then their binding to polyadenylation tail, they are very important, and their phosphorylation and dephosphorylation process actually is helping in their binding and again in their separation. Okay, so polyadenylation and circularization of messenger RNA through binding of poly A PABP to eukaryotic initiation factor 4G. So here again you can see that they can interact with each other and they can circularize. Again, this circularization of messenger RNA is again proved by the atomic force microscopy when they were visualizing the circular messenger RNA. So here you can see the circular ring-like structures, they are seen in atomic force microscopy. Okay, so the light color region is actually the proteins, okay, which are bound at the 3' end, at 5' end, and the ring-like structure is actually the RNA. Okay. So the assembly of pre-initiation complex. Initiation factors 1, 1A, and 3, they bind to 40S ribosomal subunit, and then initiation factor 2 activated by GTP and carrying the first methionine tRNA, it joins the 40S complex. So this all information is shown in this figure. If you look at this figure, you can see the first step, initiation factor 2 with the help of GTP will be bringing the transfer RNA carrying methionine, and then the small S subunit 40S, initiation factor 1A and 3, they will be bringing the small S subunit, and then messenger RNA will be actually straighten up and will be kept in position with the help of initiation factor, whole family of the initiation factors, and then the whole complex will be formed. Okay, so just see that the large subunit has not joined this whole complex. So messenger RNA scanning, AUG recognition, and then ribosome completion. So the pre-initiation complex, it travels downstream along the 5' UTR of the messenger RNA until it arrives at the start codon AUG, and it recognizes it through interaction with the EIF2 GTP tRNA complex. So just remember that we studied the function of 16S ribosomal RNA in case of prokaryotes, and its function was to interact with the Schindler-Gano sequence. So similarly, within the eukaryotic system, 18S ribosomal RNA does have a region that can interact with the 5' UTR of the messenger RNA to recognize it. Okay, so then GTP is hydrolyzed by IF5. The pre-initiation complex unravels the 60S subunit, binds to the 40S subunit, and translation begins.
Excuse me, मैम
जी
मैम हमने प्रोकैरियोट्स में 16S RNA पढ़ा था ना, जो हेल्प करता है बाइंड करने में मैसेंजर RNA के, तो जो यूकैरियोट्स में तो टोटल राइबोसोम 80S है, तो फिर इसमें 40S वाला जो है वो करेगा हेल्प?
टोटल 80 है, लेकिन इसमें जो कंपोनेंट्स है ना, स्मॉल एंड लार्ज दोनों उसी तरह, यानी 60S और 40S है ना। इसका जो लार्ज है वो 60S है और जो स्मॉल है वो 40S है। अब स्मॉल जो है 40S, उसमें 18S राइबोसोमल RNA है, ठीक है, और इसका फंक्शन जो है वो वैसा ही है जैसे 16S राइबोसोमल RNA स्मॉल सबयूनिट प्रोकैरियोटिक सिस्टम में था। ठीक है। So disassembly of the pre-initiation complex upon recognition of the start codon. So here you can see once the start codon and anticodon, they are being recognized, then all the initiation factors, they are disassembled, okay, disengaged from the this complex, and then 60S ribosome with the help of GTP will be entering into this complex. Okay, so the ribosome has two sites for tRNA called P and A site. Right, this is again the same information. The initial tRNA with attached amino acid is in the P site. New tRNA corresponding to the next codon on the messenger RNA binds to the A site. The ribosome catalyzes a transfer of the amino acid from P site onto the amino acid at the A site, forming a new peptide bond. So the ribosome then moves down one codon, and the new now empty tRNA at the P site is displaced off the ribosome, and the tRNA that has growing peptide chain on it is moved from A site to P site. The process actually is repeated. The transfer RNA at P site holds the peptide chain, and new tRNA binds to the A site. The peptide chain is transferred onto the amino acid attached to the A site tRNA, and the ribosome moves down one codon, displacing the empty P site tRNA. So this is the whole process that is repeated again and again. So you can see, again, this is depicted in the figure as well. So the elongation cycle, the translation starts with AUG start codon positioned at the A site of the ribosome. As the ribosome continues to scan the messenger RNA, the AUG tRNA MET complex is translocated to the P site of the ribosome. A new AA tRNA arrives at the vacated A site, courtesy of elongation factor. You know, the elongation factors will be bringing every next new tRNA, okay, carrying the amino acid. And elongation factor 1A requires GTP for activation. So here you can see the elongation cycle in eukaryotes. Successive amino acids are brought to the vacant A site by cognate tRNA bound to elongation factor 1A dot GTP. Okay, so here you can see in the P site, tRNA having the peptide chain is attached. A site will be actually the EF1A will be bringing the next amino acid and tRNA to this site. And then this elongation factor undergoes cycle of GTP and GTP conversion, okay? And then once this transfer RNA is actually well positioned at A site, then elongation factor will be released and GTP will be degraded into GDP and inorganic phosphate, and then the cycle will be repeated. So again, the same thing is repeated. The AUG tRNA is now at the P site, and the next available codon of messenger RNA is now at the A site, and the cognate amino acid tRNA elongation factor 1A GTP binds to it. The ribosome catalyzes a peptide bond between the methionine at the P site and the new amino acid. However, its tRNA is still at the A site. So elongation factor EF2 GTP enters the ribosome, pushing the new tRNA into the P site and deacetylated first tRNA into the exit site. So in the next cycle, this tRNA will be ejected from the ribosome. So automatically, this is a kind of process in which, you know, it's being done in a as machines are working. Okay, so from one side, one thing is entering, and from other side, another thing is being removed, right? And at the top, the peptide chain is being synthesized. And during this process, GTP is hydrolyzed, and elongation factor 2 GDP leaves the ribosome, and cycle repeats many times. Then comes the termination of translation. So again, as we read in case of prokaryotes, we were having release factors because there is no amino acid for stop codon. So releasing factors are being used to terminate the translation process. Eukaryotic release factor 1, it structurally mimics tRNA that is bound to eukaryotic EF1A.GTP. So eukaryotic RF1 fits into the ribosomal A site, where it recognizes the stop codon. It then releases the completed polypeptide by catalyzing a nucleophilic attack on the ester bond between the peptide and the P site tRNA. So it actually breaks the bond between the tRNA that was carrying the whole peptide chain, okay, and then the peptide chain is released. So the catalytic activity of ERF1 is stimulated by the GTP bound form of another releasing factor RF3. So another releasing factor RF3 is helping with the help of GTP to finish this process. So the mimicry of tRNA by releasing factor is shown here. You can see the structure of on the right side in the yellow color, whole yellow, is yeast tRNA, phenylalanine, right? And its parts are shown like anticodon loop, T-stem, and CCA, and then anticodon bases. And in the left side, you can see the human eukaryotic RF1, the release factor 1, the structure. So you can see the overall shape resembles very much with the transfer RNA, because that's why it can fit into the A site of the ribosome. And different domains are shown, and they are quite, resembling with the transfer RNA. So domain 2 on RF1 recognizes the stop codon, and domain 3 catalyzes the hydrolysis of the completed polypeptide from the P site transfer RNA. So 3 codons are called stop codons, right? They code for no amino acid, and all protein coding regions end up in a stop codon. You know, the open reading frame actually completes when we reach at a stop codon. If there's no stop codon, then definitely the protein synthesis will be halted, or it will be aborted. When the ribosome reaches a stop codon, there's no tRNA that binds to it. Instead, proteins called release factors, they bind and cause the ribosome, the messenger RNA, and the new polypeptide to separate. The new polypeptide is completed. Note that messenger RNA continues on past the stop codon. The remaining portion is not translated, and that is the untranslated region. Okay, so once the proteins are synthesized, they get post-translation modifications, okay? So new proteins, polypeptides, they usually fold themselves spontaneously into their active conformation. However, some proteins are helped and guided in the folding process by chaperone proteins. So many proteins have sugars, phosphate groups, fatty acids, and other molecules covalently attached to certain amino acids. So most of this is done in the endoplasmic reticulum. And many proteins are targeted to specific organelles within the cell. Targeting is accomplished through signal sequences on the polypeptide. So it means that proteins themselves, they have the motifs or sequences which actually guide them to reach to their destination. So in case of proteins that go into the endoplasmic reticulum, the signal sequence is a group of amino acids at the end terminal of polypeptide, which are removed from the final protein after translation. Okay, so this was end of translation process. There is a short presentation that I would like to I will be sharing with you.
मैम ये है हमारे पास
जी जी वो जो प्रोटीन सिंथेसिस है ना साइटोप्लाज्म. Can you see this presentation? Protein synthesis in cytoplasm and endoplasmic
yes
reticulum. Okay. So this is important for you people to understand that protein synthesis, how it's taking place and within the cytoplasm. So proteins have different locations, like the final destination of proteins is different within the cell and it can go outside of the cell, right? For example, the extracellular space, plasma membrane, or the membranes and lumen of several organelles involved in the vesicular trafficking, such as Golgi apparatus, endosomes, and lysosomes. So many proteins will be part of the plasma membrane and of other membranes. Transmembrane protein, for example, right? So furthermore, rough endoplasmic reticulum needs to synthesize proteins for itself also, which are known as resident or constituent proteins. You all know that rough endoplasmic reticulum is a whole network of membranes which processes the translation process, right? And then it also modifies the proteins, and then it sends proteins to the next organelle, that is the Golgi apparatus, right? So rough endoplasmic reticulum is important, you can say, organelles, right? And it has lots of different streets and, rooms inside it, and which perform different modifications, right? Which can actually modify the protein, for example, which can help in folding as well, okay? So this is one thing. And another thing is that each protein actually should know its correct destination in the cell. And this is accomplished by signal peptide, right? The short sequence of amino acid or specific modifications of the protein, which work as postal addresses. For example, endoplasmic resident proteins, they contain a 4 amino acid sequence close to the end carboxyl group, which actually make these proteins to reside within the endoplasmic reticulum. Okay, so this slide actually tells us a kind of a general mechanism that within the cytoplasm, protein synthesis is actually started, right? But after first 20-25 amino acids are being synthesized, if the protein, the peptide chain, it contains the signal peptide, then that protein has a different destination. But if the protein is not having the signal peptide in its first 20 or 30 amino acids, then that protein continue to synthesize on the within the cytoplasm, right, on the free ribosomes. But if it's having the signal peptide, then it has to be sent to the endoplasmic reticulum. Okay, so ribosomes remain free in the cytosol if they are synthesized. The polypeptide is destined for the cytosol or for import into the nucleus, mitochondria, or chloroplast, or peroxisome. So when the polypeptide is complete, it is released from the ribosome and either remain in the cytosol or it's transported into the appropriate organelle by a post-translational import. So polypeptide uptake by the nucleus occurs via the nuclear pores using a mechanism different from that involved in the post-translation uptake by other organelles. So you can see in the picture that the protein which are synthesized on the free ribosomes within the cytosol, they are either folded by themselves because they have the whole capacity to fold, they don't need chaperones, and then they can they stay in the cytosol, or they can be imported into nucleus, into mitochondria, into chloroplast, or to other organelle. But the ribosomes attach to endoplasmic reticulum membranes if they are synthesizing polypeptides destined for the endomembrane system or for export from the cell. So as synthesis continues, the newly forming polypeptide is transferred across the ER membrane by co-translational import. The completed polypeptide either remain in the endoplasmic reticulum or is transported via various vesicles to another compartment of the endomembrane system, right? Integral membrane proteins are inserted into ER membrane as they are made rather than being released into the ER lumen. So because this rough endoplasmic reticulum is having lumen, that is its cavity, internal cavity, and its membranes also. So there would be there would be some proteins which will become part of its membrane, and there will be some protein that will become part of the lumen of the endoplasmic reticulum. So depending upon the destination, they would be having the corresponding sequences in the structure, in the whole sequence. So then they will be destined to the Golgi complex, and then to the secretory vesicles, and then they can be incorporated into the plasma membrane, or they can be sent into the outside of the cell, right? And in case of plants, they can be sent to the cell wall, they can be sent to the apoplastic region of the extracellular space. Okay, so this process is just showing us that when the first 20 or 25 amino acids are being synthesized and they are exposing the signal peptide, right? Then the signal peptide is being recognized by signal recognition particle, SRP, and this signal recognition particle will be bringing the whole messenger RNA and ribosome towards the endoplasmic reticulum. And then on the endoplasmic reticulum membrane, there is a whole system, okay? There's a translocon receptor, there's another receptor where this signal recognition particle will be bind, and then the protein will be sent within the lumen of the endoplasmic reticulum with the help of this translocon, okay? So its name is like that, translocation for translocation. And then the rest of the translation will be taking place within the lumen of the endoplasmic reticulum. And here you can see there is within the part of this membrane of the endoplasmic reticulum, you can see the enzyme called as peptidase that will be releasing the signal peptide here, and then the protein will be folded, or even within the lumen of endoplasmic reticulum, there are chaperones which can either further help in the folding of the proteins. Okay, so this figure is just showing us the types of modifications which are being done on the proteins, right? So there's a whole list of modifications: phosphorylation, glycosylation, ubiquitinylation, subylation, disulfide bond formation, acetylation, lipidation, methylation, hydroxylation, etc. Okay. So this is the end of translation process. If you have any question from this part, you can ask. Any question, any confusion?
स्लाइड नंबर 22 का नहीं है।
रिपीट करें तुबा?
मैम जो आपने पहले लेक्चर दिखाया था उसमें स्लाइड नंबर 22 को एक्सप्लेन करना।
इसकी बात करें टर्मिनेशन ऑफ ट्रांसलेशन की?
यस मैम।
हाँ,
यस मैम।
ये है टर्मिनेशन ऑफ ट्रांसलेशन, जिसमें लिखा है रिलीजिंग फैक्टर्स दे आर इन्वॉल्व इन टर्मिनेशन ऑफ द ट्रांसलेशन. So release factor 1 actually structurally mimics tRNA. देखिए स्ट्रक्चरल मिमिक्री ये होता है कि कोई चीज, मिमिक्री किसे कहते हैं कि जो आप किसी तरह की एग्जैक्टली कोई चीज हो, ठीक है, चाहे किसी आवाज की मिमिक्री निकाल करें या किसी के स्ट्रक्चर की मिमिक्री करें ना, तो रिलीज फैक्टर्स जो हैं वो स्ट्रक्चरली मिमिक करते हैं tRNA को, ठीक है. और आप उस टीआरएनए को जो कि इलॉन्गेशन फैक्टर 1A जिससे बाउंड किया हुआ हो ना, तो उसके साथ मैच करते हैं बिल्कुल ही, और फिर वो इजीली फिट आ जाते हैं ए साइट पे, और वहां पे वो रिकग्नाइज करते हैं स्टॉप कोडॉन को, और फिर वो रिलीज करते हैं कंप्लीटेड पॉलीपेप्टाइड बाय कैटालाइजिंग अ न्यूक्लियोफिलिक अटैक ऑन द एस्टर बॉन्ड बिटवीन पेप्टाइड एंड द पी साइट टीआरएनए। आप देखें कि पी साइट टीआरएनए आपको इसमें देखना चाहिए, ठहरें, हमने जो प्रोकैरियोटिक सिस्टम पढ़ा था ना, उसमें पूरा वो शो किया हुआ था ना कि पूरा बॉन्ड जो पेप्टाइड बॉन्ड है वो कहाँ पे बन रहा है, और फिर ट्रांसफर आरएनए जो अटैच है जिसने कैरी किया है अमाइनो एसिड, ठीक है, तो वो बॉन्ड जो है उसको ये ब्रेक करता है, उसको भी अटैक करता है, वो ब्रेक हो जाता है, ठीक है, और उसके बाद फिर पेप्टाइड जो चेंज है वहां से रिलीज हो जाती है, ठीक है, और उसकी कैटालिटिक एक्टिविटी जो है वो जीटीपी बाउंड फॉर्म जो है RF3 की उससे स्टिमुलेट होती है, ठीक है। क्लियर हुआ तुबा?
ओके मैम।
कोई और क्वेश्चन है तो कर लें इसमें, और अगर नहीं है तो फिर जो फर्स्ट ग्रुप है आप लोग अपनी प्रेजेंटेशन शेयर करें इस पे।
मैम
जी
मैम आपने कहा था थोड़ी देर पहले कि यूकैरियोट्स के अंदर स्टॉप कोडॉन नहीं होता, रिलीजिंग फैक्टर्स आते हैं और ट्रांसलेशन को स्टॉप करते हैं।
हैं? बिल्कुल नहीं कहा मैंने।
ठीक है मैम,
देखा है, मैंने ये नहीं कहा। देखो स्टॉप कोडॉन तो होगा ही होगा ना, आपकी ओपन रीडिंग फ्रेम कंप्लीट ही नहीं हो सकती जब तक स्टॉप कोडॉन ना हो, ठीक है। स्टॉप कोडॉन होगा, स्टॉप कोडॉन के लिए कोई अमाइनो एसिड नहीं होता, ठीक है, ना ही प्रोकैरियोट्स में और ना यूकैरियोट्स में। तो चूंकि कोई अमाइनो एसिड नहीं है उसके लिए, तो वो स्टॉप कोडॉन है, तो वहां पे अब कोई ट्रांसफर आरएनए तो आएगा नहीं, ठीक है ना, क्योंकि कोई अमाइनो एसिड नहीं है। तो ट्रांसफर आरएनए की जगह फिर ये रिलीजिंग फैक्टर्स आते हैं जो कि ए साइट पे फिट इन आ जाते हैं, ठीक है, और फिर वो न्यूक्लियोफिलिक अटैक कराते हैं पेप्टाइड बॉन्ड, ताकि जो पी साइट पे जो अटैच है ट्रांसफर आरएनए आखिरी वाला जिसने इनकॉर्पोरेट कराया था अमाइनो एसिड को, उसका बॉन्ड ब्रेक कराते हैं, और फिर वो रिलीज होती है पॉलीपेप्टाइड चेन वहां से। अच्छा जी, फर्स्ट ग्रुप जो है आप लोग अपनी प्लीज प्रेजेंटेशन शेयर करें इस पे।
मैडम यहाँ सिग्नल वी के आपको मैंने ईमेल किया, व्हाट्सएप के थ्रू भी डाला है, जरा आप शेयर कर लीजिए।
ईमेल की है? अच्छा
जी जी मैडम।
अच्छा, ईमेल तो आपने मुझे किया, मैं देख लेती हूँ। अच्छा, नेक्स्ट आप लोग जो है ना अपनी अपनी प्रेजेंटेशंस को ज़रा प्लीज़ प्रॉपर नाम के साथ कर दें, जैसे जो आपकी प्रेजेंटेशन का जो टाइटल है उसके साथ करें उसको, ताकि ठीक है, प्रोफेशनल तरीका है ना कि आपने उसको उस तरह से करना है।